Okay, so this is key assessment 3, paper 1 for the upper 6th, and yeah, I can't see what the heck you can see. Anyway, it'll be interesting. I'm just going to move this over a little bit so we can read through the question, and I'll have to leap to my feet and move it back. Get lots of exercise doing this. Right, in Wales, we're looking at 12,000 to 24,000 kilometres of rivers are estimated to be acidified. So I'm thinking pH is probably quite important here. Forests capture acidic pollutants from the atmosphere and release them into stream water. How kind. So, a group of students investigated the effects of acidification, that's pH, on biodiversity. So pH is our independent variable, and biodiversity is presumably our dependent variable. Kick sampling. Now, this isn't necessarily on your syllabus, so they're going to tell you quite a bit about it. It was used to compare the biodiversity of a moorland stream. It tells you the pH, and a forest stream, pH 5. Remember that is lower and therefore more acid. So we've got a net 30 centimetres deep and 25 centimetres across. It's told us something about the net. You can see that the water's going that way, the net's downstream. And here we've got somebody in waders, not suspenders. It looks like it, but they aren't. So, kick sampling method. Place the bottom edge on the stream bed downstream, kick the stones just above the stream, disturbed material drifts into the net, empty the net into a tray with stream water, identify and count, return the invertebrates gently to the stream. That's kind of ethical issue there. So they've told you how to go about it, they've told you what they've been investigating. Leaps to feet again. Okay, let's have a look at the questions then. So, both streams were sampled in shallow, fast-flowing regions. So, that's something that they have controlled. Samples were taken from the same width and water depth, again controlled. Similar stony stream beds, controlled. 2 other variables that would need to be as similar as possible. So they've told you sort of 4 control variables. So, what else would need to be the same? So I'm thinking, standing in a stream, the faster the current, perhaps the more stuff will get knocked into the net, and the better, better your data will be. So that might be important to keep the same, as similar as possible. Temperature is always a good one. Maybe oxygen concentration, because that directly affects the organisms in the stream. How much light there is, whether it's surrounded by vegetation on the banks, perhaps what time of year you would do it. So let's go for, I'm liking time of year, because if that's different you're going to find more stuff in summer than winter presumably. So I'm going to put the time of year the sampling was done. And I'm liking, what else do I like? I quite like the flow rate of the river. Be no use doing one in a slower flow and one in a faster flow. When kick sampling, so this is actually talking about the sampling technique now, state 2 factors that need to be controlled. So I think this is pretty easy. So the time you kick for, or the intensity. So time and intensity of kicking. I think probably a mesh size is probably quite important. It's only told you the dimensions, so your mesh size of your nets will need to be the same. Maybe how far away you are from the bank, but we only need 2. Same area that you sample, you know, you always sample the same, a metre squared or whatever. To improve the accuracy of species identification, it was suggested that the specimens collected could be preserved in alcohol and taken back to the laboratory. Oh, good idea. Discuss why this is considered to be unethical. So remember I said that returning the invertebrates gently to the stream would be an ethical thing to do. So if the animals were to be preserved in alcohol, they would be killed. That's always going to be an ethical issue. And this might reduce biodiversity stream. So you're not putting everything back as you found it. Okay. Hurrah. Oh Lord. So luckily I've got my trusty calculator, so that's good. So we don't have to use too much that's from the other side of the paper. Anyway, 5 kick samples. So we've got the results, moorland stream 6.5. We've got the species, caddisfly, stonefly, wandering snail. And so that's our less acidic, that's our more acidic. We've got a total of 84, we've got a total of 48. The students collected invertebrates at sites chosen at random. Explain the importance of the sites being chosen at random. So this is quite a frequently asked question. So this is to remove bias. It's always to remove bias. It's like ethics is always about killing things. This one's always about removing bias. And that can be a sort of fairly unconscious bias by a sampler. So there's 2 reasons why the result values obtained might be an underestimate. So obviously you're going to have things that are not caught by the net, some things will drift off around the edges. So some organisms miss the net. That are not caught. Some might not be dislodged. You're never going to collect absolutely everything out of that particular stream bed. So they have to let go of their rocks before you can catch them. All you're doing with kicking is kind of... I can't spell dislodged. Oh yeah, I can. You might not kick the entire area, something might be just too small, some things you might not be able to identify. I'm just going to leave this paper again. And then we've got to calculate an index of diversity. Luckily, they've done quite a bit of the calculation for you. So, this is for the moorland stream. So this is our one. I'm just going to write the pH in there, just to remind me. So this is the less acidic. Might need to use that later on. Okay, trusty calculator. So if big N is 84, big N minus 1 must be 83. I need to multiply those together. 84 times 83. Wow. Oh, I don't know whether I've got it set to the right number of decimals. Come on, calculator, cooperate. You might need a bit better calculator than this in your exam. I suggest that you, I'm going to use this one, that you avail yourself of one of these. I find them pretty annoying, but you can put a lot of stuff in here. So we've got 84 times 83, this will get me a better answer, I'm sure. Oh, same answer. 6972. And then we need to add up, so sigma means add up. This calculation, the n minus 1's already been done for you, so this is a straightforward adding exercise. And the reason I quite like these is it actually, so you can't really miss a number out, you can always check before you hit answer that you've actually added up the right number of numbers. You haven't made a mistake. Plus 462 equals 2098. Right, I'm just going to do that calculation again. This is a really good idea because it's just a little check to self. Have I written the right numbers down? 6972. Got the same answer. Obviously sometimes I don't get the same answer. If I don't get the same answer, I do it again. Write down both answers that I've got and then do it again and see which one is right. It's 2 plus 1, 4, 8, 2 plus 462. Got all the answers on that. 2098. I'm a genius. Simpson's diversity index. So now we're doing, I need to remember to do that. It's really easy to just do this and leave it at that. So I'm doing that number divided by that number, then I'm going to make it a minus and then I'm going to make it a 1. There's probably some really clever way of doing that on this calculator in one step. I just don't know what it is. 6972. So that bit, so that will give me 1 minus 0.3. I'm only going to go to 2 decimal places, my life's too short. So that would be 0.7, wouldn't it? Which would be brill. Just do that again. So 2098 divided by 6972. Yeah, perfect. Okay. The index of diversity for the forest stream is 0.4, so this is a lower diversity. With reference to the results table on page 4, that's this one, explain how you could deduce that the forest stream is less diverse than the moorland stream without needing to calculate the diversity index. Well, okay, we've got a couple of species missing there and there, so there are fewer species. In the forest stream. So what have we got? What have we got? We've got not much species richness. We've only got really few numbers. When you think that's 22, 2, we've got 8. We've not got many swimming beetles. With no freshwater shrimps, with no wandering snails, but we've dealt with that. We've got loads of stonefly, but not very many case caddis. So we've got less species evenness, I suppose, is the good way to put it. I'm not sure that's how you spell evenness. Might have 2 N's in it. So what have we got most of? Stonefly nymphs. Stonefly nymphs dominate. You think 37 out of the 48 organisms that you've found are stonefly nymphs. It's pretty much, oh, there's another one. Oh, there's another one. So, the student concluded that forests reduce species diversity in streams. You would think so, wouldn't you? How could we improve confidence? you could sample a larger area. Or study more than 2 streams. This is all about just getting more data that supports your conclusion. Suggest how forest managers could use this information to increase biodiversity. So I'm thinking the forest stream has got, I'm just going to draw a Christmas trees on each side, an little stream running down the middle. So obviously in a forest you don't want to cut down all the trees, but possibly you could cut down the ones right next to the stream. If you got rid of those, then you would have a stream, a bit of space, and then some trees, so they might get more light. I suppose if you're thinking it's pH, you could probably causing a difference, which is probably, to be honest, caused by conifers in the first place. But you could possibly add lime into it like they do to soils. So suggest cut down the trees next to the stream, or neutralise the stream with lime. I'm thinking if you've got things that are tolerant to acid in there, possibly you don't want to be getting rid of those by adding lime, but anyway, who am I to argue? Oh, can't really see that. Ah, there we go. Ta-da!